Hello everyone, we are Group 9. We are going to talk about antiserum. Antiserum is not a familiar word, but you have probably heard of serum. On the next slide, I will first talk about serum. Serum is plasma from which blood coagulation factors such as fibrinogen have been removed. Serum can be obtained in upper layer by centrifuging the blood after collecting it and waiting for it to coagulate. So what is antiserum? An antiserum is literally a type of serum that contains polyclonal antibodies. In the figure, the Y-shaped is the polyclonal antibody. Polyclonal antibodies are antibodies against a specific antigen that come from different types of B cells. How do polyclonal antibodies react when antigens are actually introduced? Polyclonal antibodies bind to antigens in this manner. The circles, triangles, and squares on the antigen are called epitopes, and the antibody binds to these sites. However, each antigen binds only to the epitope of the same color as the binding site of the antibody. Each antibody recognizes different epitopes of the same antigen. This binding neutralizes the antigen and prevents it from causing harm to the body. Also, due to the polyclonal antibodies, the antiserum strongly reacts with specific antigens. So how to obtain antiserum? Antiserum is obtained mainly from an immunized animal. First, inject a specific antigen into an animal to produce antibodies in their blood. Inject the antigen several times to make the horse immune to the antigen. Then the horse's blood is collected and the blood is centrifuged to obtain antiserum. Finally, we will talk how antiserum is used. Antiserum is used for passive immunization and administered by injection into the human body. Antiserum is mainly used in the treatment of snake venom. In addition, since antiserum derived from horse blood is administered to the patient, one should be aware of allergic reactions, as mentioned earlier. Antisera are used as a temporary treatment for new viruses for which no vaccine has yet been developed. Antiserum is still used against the Ebola virus and was used against in COVID-19. When pandemics occurred, Antisera obtained from people who had once contracted that prevalent virus and were cured were given to patients. This concludes my presentation. Thank you for your attention.